Welcome to the 2021 American Diabetes Association Pathways to Stop Diabetes Symposium. I am Dr. Ruth Weinstock, ADA President of Medicine and Science. We are here today because we are united in our commitment to confront the burden of diabetes. The impact of diabetes and its complications continues to grow, placing serious economic, emotional, and physical burdens on millions of people. 1 in 10 Americans is affected by diabetes, and nearly half of all American adults have diabetes or prediabetes. Every 21 seconds, someone in the US is diagnosed with diabetes. To put into perspective, in the short 5 minutes we've been together, 14 new diabetes diagnoses have occurred, and by the end of this session, that number skyrockets to 343. Research, through the innovative and dedicated investigators who commit their lives to progress and discovery, can be the transformational change that is vital for people living with diabetes. The Pathway to Stop Diabetes program is the premier example of how we can successfully work together to drive results. The vision of the Pathway to Stop Diabetes program is simple, but it's revolutionary: to identify and support a new generation of brilliant scientists at the peak of their creativity, provide them with freedom, autonomy, financial stability to perform the work, and professional resources to set them on the road to making breakthrough discoveries. We believe that if we could bring a group of the best and brightest scientists, the truly gifted breakthrough innovators, into a diabetes community, encourage collaboration, and provide critical mentorship and support, we could lead the charge in accelerating and providing sustainability for the future of diabetes research. As this grantee stated, with this award, I have expanded my human evolutionary genetics research into a new area, the study of genetic risk factors for type 2 diabetes in people of African ancestry. The guidance I receive from mentors has been particularly helpful for learning novel approaches for studying risk factors for diabetes. The Pathway to Stop Diabetes initiative was developed with the guidance of the association's Research Policy Committee and organization leadership, and that support continues today. The outstanding and dedicated Pathway Mentor Advisory Group, many of whom are in attendance today, provide a solid foundation for awardees, offering both scientific guidance and professional advice, creating a challenging and collaborative environment for transformational science to thrive. So we thank all of these amazing and generous volunteers for your continued support, leadership, and commitment to the Pathway program, and to your exceptional mentees. The Pathway program was launched and continues to be sustained through generous financial support from a number of corporations, individuals, families, and foundations. And I would like to take a moment to recognize some of those donors. 3 founding corporate sponsors made it possible for us to launch the program in 2013. We thank Eli Lilly and Company, Novo Nordisk, and Sanofi for this foundational support. Founding corporate sponsor Novo Nordisk. Novo Nordisk has contributed $15 million to Pathway to Stop Diabetes to date. I'd like to recognize founding corporate sponsor Sanofi. Sanofi has contributed $12.25 million to Pathway to Stop Diabetes to date. And please join me in recognizing founding corporate sponsor Eli Lilly and Company. Lilly has contributed $8.5 million. And we thank AstraZeneca for 4 years of corporate sponsorship totaling $8 million to date, and as a 2019 initiator sponsor. We are now pleased to recognize Merck for continuous corporate sponsorship since 2014. Merck has contributed $3 million to date. In addition, we thank our corporate contributor sponsors, Abbott, BD, Dexcom, Janssen, and Pfizer. Not only do these sponsors provide critical financial support, but by networking with the awardees through events such as this symposium, and by engaging our Pathway scientists in presentations and collaboration opportunities, They help ensure that pathway discoveries are translated to the next level where they can help people with diabetes. Please join me in thanking all of them for their vital partnership. Pathway program has also been sustained by generous donations from individual philanthropic supporters. We are pleased to recognize our visionary supporters, including Dr. Karen Talmadge, past chair of the American Diabetes Association Board of Directors, and her husband, Dr. John Fiddes, Peter and Linda Cummin, as well as Peter and Valerie Capanez, and Don and Arlene Wagner. These individuals had contributed each more than $1 million to Pathway to Stop Diabetes. And thank you to our accelerator supporters who have donated between $100,000 and $1 million. We would especially like to recognize Dr. Orville Kolderman and Gail Lorenzi Kolderman for their sustaining support of the ADA's work. Finally, our initiator supporters who have donated between $10,000 and $100,000. Together, our corporate sponsors and philanthropic supporters have contributed more than $53 million for Pathway to Stop Diabetes. Since the program launched in 2013, we have funded 7 classes of Pathway scientists, a total of 34 to date. They are bringing unique approaches and ideas to solve diabetes problems. Today, as we reflect on the progress that has been made and look to the future, I think that you will agree that together, the association, volunteers, sponsors, and donors have already made tremendous strides to improve the lives of people with diabetes. But there's a lot more to be done. We want to thank all of you here today for your dedication and commitment to Pathway. This program is extremely important to us. By supporting young junior investigators and innovative, bold ideas, it not only leads to discoveries that will help people with diabetes better manage their diabetes, and hopefully lead to new prevention strategies and get us closer to a cure, but it helps support the future of diabetes research by launching the next generation of diabetes researchers. But we still haven't met our vision to support 100 Pathway awardees. Funding is critical to the sustainability of the Pathways program, and we can't do it alone. I would now like to introduce Dr. Chris Newgard, Director of the Duke Molecular Physiology Institute and the Sarah W. Stedman Nutrition and Metabolism Center at the Duke University Medical Center. Dr. Newgard is a world-renowned diabetes researcher and is also the chair of the Mentor Advisory Group of the Pathway to Stop Diabetes program. Chris will be moderating the symposium today. Please join me in welcoming Dr. Chris Newgard, and thank you all, and please enjoy this symposium.
Thank you very much, Ruth. It's my pleasure to chair today's session and to serve as the current chair of the Pathway Mentor Advisory Group. I've been part of the Mentor Advisory Group since the inception of the Pathways program in 2013. And let me just say to you quite simply that it's been the most substantive and impactful program that I've been involved with in my 30 years of involvement with the American Diabetes Association. And I think other colleagues on this Zoom, on this call, that are members of the Mentor Advisory Group would agree with me. And many of us have been with the program since it started because of the impact we have felt. Next slide, please. And what are some of the granular metrics of that impact? As Ruth said, we have funded 34 out of a goal of an ultimate funding of 100 scientists and research projects since 2013. But I just want to give you a sense of the competitiveness of the program. Those 34 have come from more than 700 applications. So the Mentor Advisory Group not only serves as direct one-on-one advisors, which is really fun and satisfying, but also serves as the grant reviewers for this program. And I can tell you every year when we meet with often more than 100 applications for which we can fund only 3, 4, or 5 applications, the competition is intense and the discussion is intense. And we walk out of that meeting generally feeling like we have found something really innovative and creative in every one of the applications that we fund. Another metric that speaks to quality is that one of our categories is the initiator category. This is for the youngest group of investigators. It provides 2 years of funding for postdoctoral fellowship with the intent of continuing to fund as those postdocs get promoted to faculty positions. And among our historical initiator awards, 100% of our awardees have graduated to independent faculty positions within the first 2 years of the award. And for those of you who think that universities are giving out faculty positions, that's not the case at all in this difficult economy. And we think this is a really sterling record of achievement for our youngest group of awardees. We've had 170 plus publications in high impact scientific journals, and that's a growing number. Some of our awardees are relatively new to the program and are just really beginning to hit their stride and realize their potential for publications. And I want to thank Kylie Goldstein, who is the ADA organizer of the Pathways program currently, for assembling all those publications in a list for me. And I want to tell you that these are truly the snooty journals of our field where our awardees have been publishing, really the top level journals. There have been 3 startup companies founded, built on 13 patent applications, and a crazy number of invited presentations and lectures given by our awardees. Next slide, please. So here's a picture of all 34 of the awardees since 2013, organized from top to bottom in seniority. So those in red boxes with asterisks are those that are graduates of the program, came in, finished their 5 or 7 year term, and have moved on. And I can guarantee you that every one of those graduating awardees would tell you the program has been highly impactful and in many cases transformational for the research. And one of the graduates will be featured today, Marie-France Hivert. Now, we could have chosen any one pictured here to represent the program today. Everyone is doing fantastic work, but we did the following. We did not want to have anyone that spoke last year speak. We chose not to take our senior awardee category, the Innovator Award, into consideration this year in favor of our younger categories. And we also wanted to get a distribution across topical areas spanning all of diabetes research. So Marie-France Hivert will speak about transgenerational risk of diabetes. You can call that pre-diabetes, if you will. Philip White will speak about novel metabolic control mechanisms as it relates to pre-diabetes. Paul Cohen will speak about the myriad new roles of adipose tissue in regulation of metabolic biology, very relevant to obesity and type 2 diabetes. And Judith Agudo will finish off about protecting beta cells from immune attack, very relevant, of course, to type 1 diabetes. So any one of the people pictured here could have represented us today, but we've chosen to try to get a broad topical representation. Next slide, please. So with that, we're going to move on to the presentations. A very quick logistical point. You please ask questions. We're going to do a Q&A session after each talk. The way to ask questions is via the Q&A button. You're writing your questions, and I will read out the questions and have our speakers answer the questions live after each talk. Next slide, please. And one more. And so with no further ado, our first speaker today is Marie-France Hivert. She got an Accelerator Award, which is for assistant and associate professors to really give them a catalytic boost as they're developing their faculty careers. Marie-France is at Harvard Pilgrim Healthcare, and she's going to talk about understanding pathways of fetal metabolic programming to stop the transgenerational risk of diabetes. Marie-France.
Thank you very much. So I'm going to take over the screen. And you guys can see my main presentation.
Yes.
So I would like to thank the AIDA for inviting me to talk in this year Pathway Symposium. It's really a great pleasure to be here. In my talk today, I will share with you some of the most recent emerging evidence on the links between epigenetics and gestational diabetes. But as you will see, the association may not be in only one direction. I have no conflict of interest to disclose. So many studies are suggesting that there is a transgenerational vicious cycle where adverse metabolic in utero events lead to altered fetal growth and metabolism, somehow programming the child for excess adiposity and all its metabolic consequences, mainly type 2 diabetes. In my lab, we investigate how gestational diabetes or maternal hyperglycemia can lead to this programming. One of the questions that we are trying to answer is what are the determinants explaining this transmission of obesity and diabetes across generations? One of the obvious hypotheses is it might be genetics. If mom is carrying genetic variants that increases her own risk of obesity or diabetes, then her child is also carrying part of this genetic burden. Others would say, well, it might be because of shared environment and family choices, such as diet and activity that have all been linked to risk of diabetes over the life course. But there are also studies that suggest that it might be more than genetics or familial environment. In this study of the Adam population, Dabila and colleagues used sibling pairs where one sibling was exposed to diabetes in utero because the mother had developed type 2 diabetes prior to the index pregnancy, while the other sib was born before the diagnosis of diabetes, so hypothetically not exposed to hyperglycemia during, or not the same degree, during in utero development. And this is a very clever design that control partly for genetics, but mostly for familial risk. And you can see in this graph that in the black bar, siblings who were exposed to diabetes during in utero development had a much higher BMI, especially during the teenage years. They also found that siblings exposed to diabetes in utero were a much higher risk for diabetes themselves, with an odds ratio of 3.7. So based on this and many other epidemiological studies, more and more investigators believe that exposure to hyperglycemia during pregnancy may lead to the programming of future metabolic disease, and became interested in revealing the molecular mechanism behind this programming phenomenon. Such biological mechanisms need to be malleable by environmental exposure, such as the maternal hyperglycemia here, but also durable and have long-lasting effect, leading to chronic diseases such as obesity and diabetes over the life course. And so our group and many others have now investigated how epigenetics might be one of the main molecular mechanisms involved in this fetal metabolic programming. Epigenetics is the study of variation in gene expression caused by mechanism other than the underlying DNA sequence. And specifically, most epigenetic phenomena are methodically stable and enduring, so they can influence level of expression over a long period of time. And some human studies have demonstrated that epigenetic marks may be stable over decades at specific loci, while other sites seem to be more malleable. Indeed, epigenetic phenomenon can be modulated by environmental factors such as pollutants, diet, smoking, either pre or postnatally, but they are particularly sensitive to in utero events. And this makes sense in the context of rapid cellular replication and tissue differentiation during fetal development. Classically, the two main phenomena that are considered epigenetic mechanisms are histone modification and DNA methylation. And some also include microRNAs as epigenetic regulator. And today I will focus mostly on DNA methylation since it's the most studied phenomenon in the context of fetal programming. With the recent rapid advances in technology, we now have arrays that allow us to investigate a large number of methylation sites across the human genome, with the most recent array being the Illumina methylation epic that covers over 850,000 CpG sites. Given the possibility to interrogate thousands of CpG sites using arrays, we can now conduct genome-wide DNA methylation association study at a reasonable cost in large population-based cohort. And in my lab, we use different measure of maternal hyperglycemia during pregnancy as our exposure of interest, and we have measured methylation in offspring in multiple tissues and now at different time points. To conduct our study, we leverage data and samples that we have collected in a prospective pre-bird cohort called Gen3G that I initiated about a decade ago. Between the years of 2010 to 2013, we recruited over 1,000 pregnant women during their first trimester. The characteristic of women we included were similar to women receiving prenatal care at our institution. We followed enrolled women between 24 to 28 weeks of gestation, where they all completed a 75-gram oral glucose tolerance test. And based on that test, we know that 8.4% of women develop gestational diabetes. We also measure glucose and insulin at the multiple time points of the OGTT to derive dynamic indices of insulin sensitivity and insulin secretion. We then follow the women until delivery, where we collected clinical outcomes. And once again, the characteristics of the newborns were as expected in the recruited population. We also collected cord blood and placenta samples in over 700 deliveries, allowing us to do a lot of the work that I will be presenting in the next few slides. Now focusing our attention on the placenta, it is well known that maternal glucose crosses the placenta and acts as the main source of nutrient in the developing fetus, stimulating the fetal insulin secretion as the main growth factor in utero. However, we don't know a lot about how the placenta adapt to this maternal hyperglycemia. And thus, we conducted an epigenome-wide association study of maternal 2-hour glucose post-OGTT and placental DNA methylation using the EPIC array to investigate 800,000 CpG sites. This is a summary of our main findings using a classic Manhattan plot. You can see on the X chromosome, on the X-axis, the 22 autosomal chromosome line up with each dot representing a probe on the EPIC array. And on the y-axis is the strength of association represented by the negative log p-value. And so the higher the dots, the stronger the association. The red line indicates a Bonferroni correction for the multiple testing that we did. And the yellow dotted line indicate the 5% false discovery rate threshold. We found that maternal 2-hour glucose was associated with placental DNA methylation at 4 loci of interest, with our strongest association at 4 probes near PDE4B. We also identify a signal near the TNF receptor SF1B, and interestingly near the LDL receptor, indicating the importance of lipid also in the placental adaptation. The 4th probe was near a gene called BLM. We pursued our investigation by performing RNA expression in placental tissues at all 4 loci of interest. We found that all 4 probes in PDE4B were positive. The DNA methylation at these probes was positively associated with higher expression level of placental PDE4B expression. And then we also found that methylation at both TNF receptor SF1B and at the LDL receptor probe that we had identified were also associated with expression levels of their respective gene at these 2 loci. But what is PDE4B? PDE4B is a phosphodiesterase responsible for the hydrolysis of cyclic AMP. Inhibition of PDE4B decreases TNF-alpha secretion, which is important in the context of placental biology and its role in inducing physiological insulin resistance. Animal models of PDE4B showed that PDE4B inhibition in pregnant mice can block intrauterine inflammation and delay preterm birth. PDA4B null mice have been shown to be leaner with lower fat pads, smaller adipocytes, and lower leptin levels. And chronic treatment with PDA4B inhibitors can delay the progression of diabetes in DBDD mice. More scarce human studies have shown that treatment with a PDA4B inhibitor of patient with recent type 2 diabetes can lower their glucose level. So overall, we can see that our agnostic approach did identify a gene that, if its epigenetic regulation is misprogrammed in early life, may very well lead to adverse adiposity and abnormal glycemic regulation later in life. Moving forward to analysis of maternal insulin sensitivity, we discovered that the placental DNA methylation markers may not only be part of programming of the offspring metabolic profile, but could also be acting on the maternal physiology during pregnancy, suggesting that the placental epigenome may have a potential contribution in gestational diabetes pathophysiology. And I'll walk you through this. So as I just mentioned, the placenta is well known for its nutrient transfer role, but we also know that it's a very active endocrine organ, influencing both fetal development and maternal physiology. One of the major physiologic changes that occurs during pregnancy is the dramatic decrease in insulin sensitivity, especially in the second part of gestation. Yet we know very little about the placental factors responsible for this reduction in maternal insulin sensitivity, since most of the commonly known placental hormones correlate poorly with insulin sensitivity during pregnancy. The placenta also adapts to this change in insulin sensitivity and thus may impact fetal development. And this tug of war between maternal and fetal energy requirements is often described as the maternal fetal conflict. And so to investigate further the placental adaptation or role in insulin sensitivity during pregnancy, we conducted another epigenome-wide association study, this time in relation to maternal insulin sensitivity, and measure at the end of second trimester in our Gen 3G cohort. This is another Manhattan plot representing results again, and the red line indicates the Bun-Ferroni threshold once again, and you can appreciate that we found a lot more signal here. Overall, we identify 188 CPG sites that reach our Bun-Ferroni threshold, and some of these are worth of mention. For example, we identify a probe near TCF7L2, which is the loci at which genetic variants have been most replicated for risk of type 2 diabetes, and also been identified as a loci with a genetic variant associated with gestational diabetes. Another probe was identified near the prolactin receptor, highly relevant during pregnancy. Another example is PRDM16, that is a DNA transcription regulator and key to differentiation of brown adipose tissue. And we also found a probe near KCNQ1, which is a well-known type 2 diabetes genetic risk locus. And it was shown to increase the risk of type 2 diabetes in a parent of origin transmission fashion, in line with the fact that this locus is a known maternal imprinted gene. Surprisingly, KCNQ1 was not the only imprinted gene we identified in our top hits. In fact, we identified 12 known placental imprinted genes. 9 of those were maternally imprinted, meaning the paternal allele is expressed, and 3 of them were known paternally imprinted, meaning the maternal allele is expressed, including the well-known H19 locus, which is located nearby IGF-2, which is the other major growth factor acting during in utero development, in addition to insulin. And so in this maternal fetal conflict hypothesis, the paternally expressed IGF-2 promotes fetal growth, and the maternally expressed H19 would presumably limit fetal growth to preserve maternal fuel. And this locus also included microRNA number 675, which has a putative role to limit placental growth via reduction of the main IGF receptor. Before moving to the next slide, just want to highlight this gene called DLGAP2. because in an attempt to untangle the direction of effect between the placental DNA methylation at the loci we identify and the maternal insulin sensitivity, we applied a bidirectional Mendelian randomization analysis. We were able to build a genetic risk score as instrumental variable capturing DNA methylation levels in the placenta for 131 of the CpG sites we had identified initially. And based on these analysis, we found supporting evidence that placental DNA methylation is causally implicated in influencing maternal insulin sensitivity level at 5 of the loci that we had identified, including this DLGAP2, which is one of the imprinted gene that I highlighted. Another fascinating aspect of our finding was that looking at the list of genes annotated to the 188 CpG sites, we were intrigued to find many other microRNAs in addition to the microRNA 675. We used microRNA target scan and found significant enrichment also for targets of multiple microRNAs in the list of genes we had identified. And so this suggests an important role for microRNAs in placental biology in glycemic regulation during pregnancy, either as paracrine or endocrine actors. And we're pursuing some of those hypotheses in our follow-up studies. To conclude, I hope I was able to demonstrate to you that epigenetics may be part of the programming of the offspring metabolic future, but also that placental epigenetic may participate to the maternal insulin sensitivity regulation, and thus contributing to gestational diabetes pathophysiology. Overall, we need to remember that the errors of our initial hypothesis may be opposite of what we had expected, and reevaluate our assumptions. In the future, we are continuing to push further our investigation of placental genomic regulation with our current NIH-supported project to study RNA and microRNA associated with maternal insulin sensitivity, insulin secretion, and risk of gestational diabetes. And I think a big part of why I was able to secure this big NIH R01 was based on our preliminary finding from the ADA pathway. Finally, I would like to acknowledge my research team and collaborators in Sherbrooke who've been instrumental in building and maintaining Gen3G, my colleagues in the Coral Division at Harvard. So I want to thank other colleagues at Mass General Hospital and inside and outside of Boston, including people involved in the multiple consortia that I have the chance to participate. And of course, I want to acknowledge all the funding agency, especially the ADA Pathway to Stop Diabetes program, that have been supporting our work for genetic and epigenetic studies in Gen3G for now more than 5 years. And on this, I'll be happy to take any questions.
Thank you. Thank you very much, Marie-France. That was a lovely summary. For some reason, I am not seeing questions in the Q&A, although I have one. So I'll start and see if questions do pop up. So in the beginning of your talk, you talked about the phosphodiesterase PDE4B and a particular epigenetic modification of that gene. There are many different phosphodiesterases. So what do you think gives that particular phosphodiesterase variant a specific role in this transgenerational risk of diabetes?
I don't know why this one particularly emerged from the signal, and I think that's the beauty of going a little bit agnostically with our genome-wide approaches, is that we're not putting a priori hypothesis of like which one would have a higher relevance. I think we have to remember also this is a signal with DNA methylation in the placenta. I think it still remains to be seen if that's acting at the placental level and changing some of the inflammation locally, then influencing the development of the fetus, or we did not find the same signal in the cord blood, let's say. but it could be that it's also happening in other tissue in the growing fetus. It just, in humans, it's a little more challenging to get tissue in healthy newborns to verify the level of methylation.
Okay, well, okay. Oh, now here comes some questions. Okay, from Bob Eckel. How consistent are data from murine models of gestational diabetes related to the human data?
So the challenge with gestational diabetes is for animal model is that a lot of the hyperglycemic model in pregnancy are done in animals where the diabetes is induced before pregnancy, and then the hyperglycemia occurs throughout pregnancy, and very few models are similar to gestational diabetes. And then the second challenge is that the human placenta is a very specific organ, and then other animal placenta are essentially very different. There is one colleague in Manitoba that has developed an animal model similar to what would look like a GDM, and so we're collaborating to push further some of those epigenetic findings.
Okay, great. We have time for two more questions from Steve Parker. Awesome work. It looks like your association scan adjusted for inferred cell type proportions. Have you tried to deconvolute which cell types these signals might be working through?
Yeah, so at the moment where we published or submitted these studies in the placenta, the cell type specific were not available, so we use other methods that are data driven. But now, just recently, there is a placenta specific epigenetic reference base that we could look into to see exactly which cell type seems to be driving our signal.
Okay, and last question from Steve Rich. As you've been a leader in this research area, do you know of other studies that could be available to replicate your findings?
So we're collaborating with the PACE consortium right now, and we're doing a meta-analysis using, so we just published last year one looking at GDM as a dichotomous trait, and we had 7 cohorts, and we couldn't really, there was not a strong signal, and it might be because GDM is diagnosed in so many different ways in all the cohorts. And so we had a lot of heterogeneity in the definition of GDM. Currently, we're running a meta-analysis looking at fasting glucose because that's the trait that most cohorts have. Every time we try to do replication with our more complex traits, whether it's like post-load or the insulin sensitivity, we cannot find other cohorts that have the same profile, but we're building a cohorts right now with my colleague, Camille Poe, for women who are slightly at higher risk of gestational diabetes, but where we are measuring all those dynamic indices and collecting placenta. And so we hope to be able to replicate in the future.
All right, unfortunately, we're going to have to move on. I apologize to my friend, Andy Stewart. Marie-France, please answer Andy's question by typing an answer. And we'll move on then, please, to the next presentation, which will be given by a 2016 Initiator Awardee, someone I know well, Philip White, from the Duke Molecular Physiology Institute. And his title is A New Homeostatic Mechanism for Metabolic Control. Phil.
Thank you, Chris. So I'm delighted to have the opportunity today to share the progress that I've made since 2016 on my Pathway Initiator Award. And my project, although the title is somewhat vague, builds off a long-standing observation made over 50 years ago that demonstrated that levels of branched-chain amino acids, valine, leucine, and isoleucine, as well as two aromatic amino acids, tyrosine and phenylalanine, are elevated in people with obesity. And my interest in this area really was spiked by the work of our wonderful chair of this meeting, Chris Newgard, when he published in 2009 that among a broad collection of metabolites measured in individuals with obese and lean humans, that the statistical metabolite factor that most strongly correlated with insulin sensitivity was not one related to lipid or glucose metabolism, but actually made up of these same amino acid components and called the BCAA, or branched chain amino acid related factor. And you can see here on the right side the different components that make up this factor, the three branched chain amino acids, valine, leucine, and isoleucine, as well as those two aromatic amino acids, which share the same large neutral amino acid transporter, as well as some catabolic byproducts, glutamate and C3 and C5 isocarnitines, which are produced by the catabolism of branched chain amino acids. This study was followed up by another study from Rob Gerster and Tommy Wang in the Framingham cohort, which showed that these same five amino acids, the three branched chain and two aromatic amino acids, were highly significantly associated with future diabetes up to 12 years prior to diabetes onset. And so this suggested that this association required a little additional understanding. And so we've been taking on this question together with Chris as a postdoc and now in my independent lab by looking at the overlap of branched chain amino acid homeostasis and different features of type 2 diabetes and related comorbidities, such as insulin resistance, glucose intolerance, dyslipidemia, NAFLD-NASH, cardiomyopathy, and even beta cell function. And the idea being that if we're able to determine if areas of perturbed branched chain amino acid homeostasis influence any of these features, that might represent a new therapeutic opportunity for type 2 diabetes and related conditions. And we've been able to make a number of nice contributions in this area. So what do I mean by nodes of branched chain amino acid metabolism? And so what I'm showing here in this complicated cartoon is the pathway of branched chain amino acid catabolism, simplified to show only one cell type. And what I'll have you see from this is that there are multiple levels of regulation of branched chain amino acid metabolism that can be regulated by their transport into the cell, their transport into the mitochondria, where most of the catabolism takes place, and then multiple enzymatic steps downstream. The one that my project for the Pathways Award focuses on is this rate-limiting step, which is called, the enzyme is called BCKDH, or the branched chain keto acid dehydrogenase, which is the second step in the pathway. Most of this activity systemically takes place in the liver. And what we know to be true for obesity and insulin resistance is that in the liver of obese and insulin resistant individuals, there is an increased expression of the kinase, which phosphorylates and turns off this enzyme, and decreased expression of the phosphatase, ppm1k, which dephosphorylates and keeps this enzyme active. And so BCKDH becomes hyperphosphorylated and inactive, and the intermediates start to accumulate, the branched chain keto acids, as well as the branched chain amino acids. And so we wondered whether restoring activity of this node in the liver would have any downstream effects on metabolism and some of those areas that I mentioned on the previous slides. So how do we go about doing that? Well, at about the time we started these studies, our colleagues at UT Southwestern, David Chuang and Max Wynn, had developed some small molecule inhibitors of the kinase, which bind to its allosteric regulatory site quite potently and act as strong and fairly specific inhibitors of the kinase. And we also generated an adenovirus so that we could overexpress the phosphatase and leverage the known adenotropism of the liver to get a more liver-specific intervention to restore activity of this branched chain amino acid node. And so by inhibiting the kinase or overexpressing the phosphatase, those were two orthogonal approaches to decrease phosphorylation and revive activity within the liver and determine what the effect was on overall metabolic homeostasis. So I'm showing a broad summary of the results. At the top is the effects of the kinase inhibitor BT2, and at the bottom is the effect of overexpressing the adenovirus for the phosphatase to exert basically the same effect. And you'll see the systemic small molecule lowers the branched chain amino acids a little bit more robustly than the phosphatase overexpression does, and same for the keto acids. But what I want you to note is that both of these interventions had almost equivocal effects on lowering hepatic triglycerides and normalizing glucose tolerance. And these were performed in fatty Zucker rats, which is an extreme model of obesity and insulin resistance. And the duration of these studies was only 1 week. So within 7 days of administration of this small molecule or overexpression of the phosphatase in the adenoviral vector, we were able to lower liver fat by close to 40% and restore glucose tolerance without impacting food intake or weight gain or activity levels. And so this was incredibly striking to us, especially considering the prior studies we performed with dietary branched chain amino acid restriction did not display such potent results. And so we felt that maybe because we were modulating a phosphatase and kinase, they may be influencing other molecules in addition to the BCKDH, which was their one known target. And so we've performed a phosphoproteomic study on the livers from both groups. And we felt that by overlapping these data sets, we might be able to identify the culprit for these benefits on glucose and lipid metabolism. And so indeed, that turned out to be true. There was only one phosphopeptide that was shared between the BDK inhibition and the PPM1K overexpression. And this was the serine 454, which is a regulatory serine on the enzyme ATP citrate lyase. And ATP citrate lyase is the enzyme that's responsible for converting citrate into oxaloacetate and acetyl-CoA. And of course, these are the building blocks for gluconeogenesis in the form of oxaloacetate, and also for lipogenesis from acetyl-CoA. And of course, when acetyl-CoA is converted to malonyl-CoA by the actions of ACC, that can also inhibit fatty acid oxidation. And so we felt like this may be a good explanation, at least for the effects on hepatic lipid content in our animals. And so we actually went on to look at this more deeply. And I don't have time to show all the data relating to this today, but we're able to demonstrate that indeed, increasing BDK expression with an adenovirus in a lean animal is sufficient to increase de novo lipogenesis by 2.5-fold. It does this by increasing phosphorylation of ATP citrate lyase, and that the balance of these enzymes in liver is actually tuned in to the lipogenic program. If you feed animals, refeed them with a high fructose diet, that's sufficient to induce the kinase and suppress the phosphatase, and that occurs through CHRBP. An important note on this is that the enhancer region we identified that CHRBP binds to in the BDK, upstream of the BDK gene, is not present in mice, but present in humans and rats. And so had we performed these studies in mice, we may not have seen the same thing. But this leads to a model where the balance of BDK and PPM1K doesn't only control branched chain keto acid oxidation, something that we're able to measure accumulating circulation of obese individuals, but also under the surface is eliciting a finer control on the rates of de novo lipogenesis and by virtue of malonyl-CoA production, potentially hepatic fatty acid oxidation. And so what are the implications of this finding? Well, one of them is that the branched chain keto acids in circulation may be an interesting way to get a read on fatty liver disease in humans, for which there's no active biomarker. And so we went on in collaboration with Andrei Chernoff at the Quebec Heart and Lung Institute, where they have a nice biorepository of liver biopsies taken from individuals who undergo bariatric surgery. And we were able to perform metabolomics in 288 plasma samples from individuals who had had their livers histologically graded by pathologists. And we looked at the endpoints being steatosis grade or the presence or absence of NASH. And the only two metabolite factors that are associated with both steatosis and the more inflammatory and pathogenic form NASH were indeed the keto acid from valine and the branched-chain keto acid to branched-chain amino acid ratio. And these associations withstood correction for an array of relevant factors, including the PMPLA3 genotype, as well as BMI and age and sex and all of the usual factors. So we've also looked at gene expression within livers from both a Quebec cohort and a Mexican bariatric surgery cohort in collaboration with Adriana Huertas Vasquez and Samuel Canizales at the University of Mexico. And what I can relate to you is that the expression of BDK within human liver of obese bariatric surgery patients is tightly correlated with features of steatosis and NASH, suggesting that this is not just something we see in rats, but it's true in humans. And this pathway may be something we can leverage to get an indication of whether an individual is at risk of NASH using simple plasma samples. In addition to this benefit in terms of treating NASH, we've also been exploring the relationship between elevated keto acid levels produced by the imbalance between BDK and PPM1K and the influence on cardiac function. And we know that individuals with type 2 diabetes have a much higher rate of death from cardiac disease and cardiac myopathy. And so we've been exploring the influence of elevated levels of branched-chain keto acids on cardiac function. We've been able to use stable isotope tracing to map their fate and determine that heart handles them quite uniquely. But something that was really striking to us was that if you incubate an isolated heart from a healthy animal with no branched-chain keto acids or branched-chain keto acids found in physiologic conditions, there's really no influence on cardiac protein synthesis measured by pure myosin incorporation. However, if you incubate those hearts with a perfusate buffer that contains branched-chain keto acid levels found in steatotic, obese, and type 2 diabetes individuals, you rapidly increase the rates of protein synthesis. And this is associated with a robust shift in the phosphoproteomic landscape within the heart. So suggesting that the branched-chain keto acids, which accumulate directly, whose accumulation is directly related to this imbalance between BDK and PPM1K, can be having effects in other organs due to the fact that they're not being catabolized. And we also show that branched-chain amino acid exposure to the heart also influences glucose and lipid metabolism in the heart and contributes to triglyceride accumulation. Now, while all this work has been going on, we've gotten some terrific genetic insight from other groups. Claudia Langenberg's group performed a really nice Mendelian randomization study where they looked at the association and they tried to triangulate genetic variants that associate with branched-chain amino acid levels with their effect on the risk for type 2 diabetes in very large cohorts. And what I'll have you note from this table below, which is the key finding, is that the one genetic variant or SNP near a gene that associates with all three branched-chain amino acids is actually in the PPM1K locus. And it associates with increased levels of branched-chain amino acids, suggesting that it's lowering PPM1K activity, and also increased risk of type 2 diabetes. So a simple way to model this would be that, based on what I just told you, loss of PPM1K activity leads to hepatic steatosis and potential insulin resistance and drives towards type 2 diabetes. However, subsequent studies from a couple of other groups, and I'm just showing one from Mika Alakopoulos's group, showed that actually these same SNPs that are associated with increased risk for type 2 diabetes and branched-chain amino acids are not associated with risk for insulin resistance. And they proposed a model wherein insulin resistance leads to increased branched-chain amino acids, as does this risk allele, and that in the setting of insulin resistance, branched-chain amino acids would drive towards type 2 diabetes. However, we felt there was an alternate explanation for this association, and this is work that's been undertaken by a terrific postdoc in my group, Jan de Ley, and he's been exploring whether PPM1K might independently promote type 2 diabetes risk, potentially by its actions within the islet. So is there evidence to suggest that PPM1K might be important in a pancreatic islet? And here what I'm showing you is data from the Tiger repository, where they actually have RNA sequencing data from human pancreatic islets, and it's compared to the GTEx data set, which allows us to understand expression levels across many organs. And what I'll have you note is that heart is the tissue with the highest expression of PPM1K. And within the regular GTEx data analysis, pancreas looked like it had very low expression. However, within pancreatic islets, the expression is equivalent to what we find in heart, suggesting the islets have robust expression and maybe one of the highest expressing places within the organism. Data from the Power Brisova's laboratory from their single-cell RNA-seq of human islets shows that this expression is spread amongst the main cell types of the islet, the beta cells, alpha cells, and delta cells. And working with Jeff Tessam at BYU, in some RNA that he had there, we're able to look and show that human type 2 diabetic islets have a lower expression of PPM1K than non-diabetic islets, suggesting that this may be quite relevant within the pancreatic islet. So Jan has conducted some siRNA studies, and his studies in our newly developed conditional beta cell knockout mouse are ongoing, but are largely supporting what I'm going to show here. And so here he's knocked down PPM1K and studied beta cell proliferation in INS1 cells. And you can see after 48 hours, there's already a modest reduction in proliferation. At 72 hours, proliferation is significantly reduced. At the same time, glucose-stimulating insulin secretion is increased. And these are mean plus or minus the standard deviation. And so there's a significant increase with PPM1K knockdown. Intriguingly, if we look at the gene expression profiles, and I'm just showing a cluster of relevant genes, we see that some important transcription factors, MAFA and NKX6.1, which control beta cell identity, actually have reduced expression, whereas transcription factor more related to the alpha cell increased, ARX, in conjunction with increased glucagon expression, suggesting there may be trans-differentiation taking place. And this fits a little bit with Dominic Chili's work on the causes of beta cell failure. So is this through BCKDH, the known target, or is it through something else like we've seen in liver? And so if we look at by Western blot, rather than seeing an increase in phosphorylation of BCKDH when we knock down PPM1K, we actually get a modest reduction in BCKDH phosphorylation. And we see this in heart as well in our cardiac-specific knockout mice. And we think it's because there's compensation from the kinase. And you can see here the kinase expression drops significantly when we knock down PPM1K. And if we run stable isotope tracing experiments, we can see that the labeled valine incorporates into the keto acid of valine, and they get a little bit more incorporation in the downstream product 3-Hib in conjunction with this reduced phosphorylation. But there's no major impacts on BCAA catabolism in these cells. So we felt that PPM1K in beta cells may be operating through a different network as we've identified in liver. And so we revisited our phosphoproteomics data from liver and noted that the most strongly modulated protein was in fact S6, the ribosomal protein S6 on its major regulatory phosphosites, serine 236 and 235 and 240 and 244. And so Jan felt that maybe PPM1K is not just regulating BCKDH phosphorylation, inhibiting and influencing S6 by modulating levels of amino acids, but that PPM1K might be acting as a brake on protein translation directly by dephosphorating S6. And this was based on the fact that in beta cells, we saw no evidence of regulation of BCKDH. And in our phosphoproteomics data, although we detected the phosphopeptides of mTORC1 and S6 kinase, which are upstream of PPM1K, neither modified. And if this were to be true, this would be the first identified phosphatase specific for S6. So Jan has begun to explore this. And what I'm showing here is Western blots from these same INS1 cells. You can see he's knocked down PPM1K quite nicely. There's no change in phosphorylation of mTOR, no change in phosphorylation of P70S6 kinase, but a very clear increase in the phosphorylation of S6 on both regulatory phosphosites. And this occurs in conjunction with an increase in protein translation, once again measured by puromycin incorporation. And in fact, the cells actually have more milligram of protein per cell. They're accumulating more protein, and they have the hallmarks of the initiation of an unfolded protein response, in that there's more XPP1 splicing and increased expression of BIP. So we think that knockdown of PPM1K and potentially SNPs associated with PPM1K may be promoting increased protein translation in the beta cell, which in the short term increases insulin production and secretion, as we're measuring in GSIS, but over the long term sets them up for beta cell failure. So is this mTOR independent? One way to look at this is to see if we can uncouple the inactivation of mTOR from the effects of PPM1K. So we treated these cells with rapamycin over a time course going out to 20 minutes and looked at the effects on phosphorylation. And you can see at baseline, the knockdown cells have higher phosphorylation of S6. And although after 5 minutes, the upstream signaling is completely abolished, the increase in phosphorylation of S6 is maintained throughout the duration of this experiment. We could also activate AMPK, which will inhibit mTOR C1 using ICAR, and we did this for 12 hours, so this is a longer term experiment. And you can see for the duration of the study, 4, 8, and 12 hours, there's an equivalent inhibition of signaling through P70 upstream of S6. However, the increment of increase in S6 phosphorylation is maintained throughout the time course of the study, all the way out to 12 hours. Suggesting that PPM1K is regulating S6 independently of mTOR C1. So with that, I'm going to come back to a summary. We've begun to study a new role for PPM1K in the beta cell built off human genetic studies suggesting it's associated with diabetes, but not insulin resistance. We find that glucose lowers PPM1K expression in the beta cell, and that loss of PPM1K in the beta cell leads to less inhibition of S6 phosphorylation, so increased protein translation. This results in increased insulin secretion, insulin accumulation, and also the development of an ER stress response, which may promote beta cell failure. We also get hints at transdifferentiation, and I can tell you that we think this is associated with induction of insulin resistance within the beta cells. There is a reduced ability of IGF-1 to stimulate AKT, and we're currently exploring that. So complete summary, I hope I've shown you that this new homeostatic axis controlled by the kinase and phosphatase first known to regulate branched-chain amino acid metabolism actually connects branched-chain amino acid metabolism or accumulation of branched-chain amino acids in circulation to both liver, heart, and beta cell dysfunction. And with that, I'd like to thank my team. And this is Jan, who did all the beta cell work, and Jackie, who's been working on the heart project. Also like to send out a big thanks to the chair of the session, Chris Newgard, who's my mentor at Duke, and involved throughout a lot of this work, as well as our great collaborators internally at the Duke Molecular Physiology and externally at the Quebec Heart and Lung Institute, as well as Jeff at BYU, Anna Gloin, who's helping us out with the human genetics component of understanding PPM1K, and David and Max at UT Southwestern. And of course, this work wouldn't have been possible at all without the Pathway to Stop Diabetes award. Thank you.
Well, thanks very much, Bill. That was terrific. I'm not seeing any questions again, and I don't know whether it's because the Q&A isn't working. Please send along questions if anyone has any. I'll just ask you to relate the really fascinating work on PPM1K and control of protein translation in the islet via S6 to what you found but in exposing the heart to high BCKA levels and that mode of activation of protein translation. Does that bypass PPM1K or serve in some way as a direct conduit of a change in PPM1K?
if you understand what I'm saying.
Yes. So we've made the cardiac specific knockout mice for PPM1K as well as the beta knockout. And we see the effect of PPM1K knockout on S6. And what's intriguing about PPM1K is depending on the tissue you knock it out in, you will or will not see an effect on BCKDH. For example, in the liver, you see an effect of PPM1K on BCKDH. But in both heart and islet, the sites of highest PPM1K expression, it does not appear to regulate BCKDH or BCAA metabolism. And we've done stabilized tracing both in the hearts of those mice, as well as the Western blotting for BCKDH, as well as in our islets. However, what we're doing at the moment is trying to evaluate whether loss of PPM1K sensitizes to those effects of BCKA. And you recall the BCKA didn't actually affect S6 in the heart, they affected 4-ABP1. But we're curious about whether adding BCKA in the absence of PPM1K will get this ultra protein synthesis phenotype.
Gotcha.
And so we're doing those studies right now.
Okay, so we're going to have to abbreviate this. The dueling cons, Stephen and Barbara. Steve Kahn, great work. Have you done your insulin cell studies in isolated beta cells and other endocrine cell types in the islet where you saw PPM1K expressed? That's Steve Kahn.
Yep. So yeah, great question. We've done work in both rat islets and human islets with Jeff Tessum, and they recapitulate the defect in proliferation, or the reduced proliferation increase in insulin secretion. We've just got our mice up and running because we made the flox and then had to back cross them to the tamoxifen inducible MIP-CRE. And we've begun those experiments. We've got some expression data which shows very similar transcriptional changes in glucagon and ARF after just 1 week of tamoxifen treatment. And then John Campbell in my group, who works with Dave D'Alessio, and he's all about the alpha cell, he's banging on me to get these into the alpha cell knockout. So we're going to be going there pretty soon, and it's just a matter of hands and time. But yeah, absolutely, these are great questions.
Okay, and brief answer to Barbara, please. Is PPM1K acting on the same phosphorylation site on S6 as mTOR? Does it dephosphorylate the mTOR site?
So it's acting on the same site as both mTOR and ERK P90 risk. So it's acting on all sites. So there's actually, we've measured 4, there's 5 regulatory sites. We measured the 4 main ones. It's acting on all of those sites. We're not affecting mTOR activity when we modulate PPM1K at all. It doesn't show up in our phosphoproteomics, doesn't show up in our Western blots. And we're also not affecting P90 risk or ERK. So we do think it's likely direct. We just need to get those in vitro studies run to demonstrate the direct phosphatase activity. And that's been a little bit challenging. So.
Okay, so I just want to alert you to there are 3 other questions from Stephen Rich, Maxence Natchery, and Michael Stitzel. Please answer those in writing, and we'll have to move on. Thank you very much, Phil. If we can move on to the next talk. 2017 Accelerator Awardee, Paul Cohen from the Rockefeller University, and his title, Dissecting the Role of Beige Fat in Metabolic Homeostasis. Paul.
Thank you for the introduction, and thank you for the opportunity to share my group's work with the group today. I have no conflicts of interest to disclose. So the problem that motivates my group's work is the enormous problem of obesity. And just to give you a sense of the scope of the problem, in the United States now, more than 2/3 of adults are either overweight and obese. And by 2030, it's predicted that 50% of adults in the United States will be obese. This is threatening to bankrupt our healthcare system, and obesity is now also overtaking tobacco as the leading preventable cause of cancer. As we have unfortunately seen over the past year or so, obesity is also a major risk factor for adverse outcomes in COVID-19, And finally, this is no longer just a problem of industrialized nations. Now more than 2 billion adults worldwide are overweight or obese. And for the first time in our history, diseases due to overnutrition are now more prevalent than diseases due to undernutrition. And so the focus of my group's work is to understand the mechanisms linking obesity to these diseases, which are really responsible for the morbidity and mortality. And so as we think about how this occurs, we feel that the answers to many of these questions can be found by studying the biology of fat. And fat comes in two main types, white and brown fat. These are electron micrographs showing you white fat on the left, which is specialized for energy storage. And one aspect of my lab is interested in understanding how white fat contributes to obesity-associated diseases. On the right is a micrograph of brown fat, which you can see has a rather different appearance, and rather than storing energy, dissipates energy. And we're very interested in understanding whether brown fat protects from obesity-associated diseases, and if so, how that might work. And so as we shift our focus towards brown fat, we and others have begun to ask whether perhaps in thinking about brown fat, is it actually perhaps part of a solution? And so this is a depiction of the key cellular features of white and brown fat cells. White fat cells have these large lipid droplets and are very efficient at storing energy in the form of triglyceride. Brown fat, on the other hand, has many small lipid droplets which facilitate the mobilization of lipid, and also a very high number of mitochondria. And these mitochondria actually allow brown fat cells to dissipate chemical energy in the form of heat. And the way that that's done is shown here. This is a depiction of a mitochondria, and what you can see here is the electron transport chain that generates a proton gradient resulting in the generation of ATP. And what is unique about brown fat cells is that they express a protein called UCP1, or uncoupling protein 1, which allows this proton gradient to leak back across the inner mitochondrial membrane. This generates a futile cycle, the end result of which is the generation of heat, or the conversion of chemical energy into heat. Now, it's probably the case that our ancestors evolved brown fat in order to protect themselves from hypothermia, which is a major threat to life, particularly early in life. And it also turns out that there are two types of brown fat. So on the left here, I'm showing you developmentally preformed, or what has been called classical brown fat. And this is thermal imaging, which measures heat emission. And this is a litter of newborn mice. And you can see here, one of the mice is ventured away from his brothers and sisters. And there's this very strong heat emitting signal in an interscapular location. And this turns out to be developmentally preformed brown fat. Rodents also have inducible brown fat. So mice will live very happily at cold temperatures. And if you expose the mice to cold, what you'll see is in this dissection is this tremendous transformation where fat now takes on this brown character. And if you look histologically, you see that many of the adipocytes now have features of brown adipocytes. And so this inducible form of brown fat has been termed beige fat by many in the field. And so dating back to my postdoctoral studies, I've been really interested in the physiological effects of thermogenic fat. And we and others have focused on a key transcriptional regulator called PRDM16. And using mouse models with adipocyte-specific loss or gain of function, we've been able to show that deleting PRDM16 leads to loss of thermogenic fat, more unhealthy visceral fat, increased inflammation, mild obesity, insulin resistance, a precursor to type 2 diabetes, and non-alcoholic fatty liver disease, whereas transgenic mice show the opposite phenotype. So these findings have led us to question whether perhaps we can target fat for therapeutic benefit, particularly for conditions like type 2 diabetes. And so when I started my independent group, we were incredibly fortunate to receive this Pathway Award, and this allowed us to really attack this question in a really ambitious fashion, focusing on multiple levels. And so my group tries to address this at three main levels: at a cellular level, asking what are the molecular determinants of thermogenic fat; at a tissue level, what are the cellular and molecular components that control the formation of thermogenic fat and the balance between healthy and unhealthy fat; and at an organismal level, which is the focus of my Pathway Award, in understanding how thermogenic fat communicates with other tissues. And this third point is actually the subject of a talk I'll be giving in a session later today in the meeting. And so what I want to highlight today is a new direction. But before I do that, I also want to just highlight regarding point one really unique and wonderful aspect of the Pathway program is that it allowed us to interact with a new cohort of experts and colleagues. And through the Pathway program, I was able to meet Praveen Sethupathi, another awardee, and he and I had this collaboration where we studied some of the basic molecular features that govern the identity of white and brown fat, and that resulted in a publication a few months ago. So getting back to our goals, another advantage of the Pathway program is that it really allowed us to broaden our approach. And since everything we do is geared towards bringing our findings to therapeutic relevance, we thought it was really important to, while we were pursuing these basic studies, to ask a more translational question, which is what role does brown fat play in cardiometabolic diseases in humans? And the conventional notions might have suggested that it wouldn't play much of a role. And the reason for that is because until fairly recently, it was thought that thermogenic brown fat was really only relevant in rodents and newborns as a way to defend against hypothermia. But over the past 10 to 20 years, a number of studies have really overturned that notion. And that actually started with some papers in the radiology literature in the early 2000s. What I'm showing you here is an image from something called an FDG PET CT scan. These scans measure the uptake of radiolabeled glucose by PET scan and then overlay them with CT scans to assess the imaging characteristics of the tissues that take up glucose. And these scans are typically used in patients with diagnosed or suspected cancer to track the progression of disease. And what radiologists noted dating back nearly 20 years ago is that very frequently these scans would show radiolabeled glucose uptake in these bilaterally symmetric tissues along the neck. And this was referred to in their papers as upper supraclavicular area fat. And although this was reported on, the physiological significance, if any, was not really understood until a series of studies in 2009 that really conclusively showed that many adult humans do in fact have cold-inducible brown fat. And I show you the three references here, one of which came from the group of Ron Kahn, a former member of the mentor advisory group of this group here. And so in this study, what you can see is an FDG PET CT scan done on a young, healthy subject, either at thermoneutrality or after just a few hours of moderate cold. And you can see this really robust glucose uptake in these tissues along the neck and cervical spine. And when these tissues were biopsied, they actually had the cellular and molecular characteristics of brown fat. And so over the intervening 10 years, these studies and a number of others have taught us a great deal about brown fat. But one of the major limitations is because brown fat is detected by these scans that require radiation and intravenous tracer administration, there have not been any large studies that are sufficiently powered to really, I would say, comprehensively assess the physiological consequences of this tissue. And so we, a couple of years ago, embarked on an effort to complement our basic studies in mice. And we took advantage of the fact that we are across the road from a very large cancer center where they do about 20,000 PET scans a year. And so Tobias Bescher in my lab conducted a retrospective review of all FDG PET scans done at Sloan Kettering over a nearly 10-year period. And because it is their practice to always comment on the presence or absence of brown fat in their reports, we categorized patients as either brown fat positive or brown fat negative. And then we were able to link patients' brown fat status to all of the other data in their electronic health records, both diagnoses as well as laboratory values. And so some of the descriptive findings we made matched those in prior studies. So first of all, brown fat is more common in women than in men. I'm showing you the prevalences here. And it's important to keep in mind that these are non-stimulated scans. So this is probably a great underestimation of the amount of people who may have the potential to have brown fat. Secondly, brown fat prevalence decreases with age. It's inversely associated with outdoor temperature, so that scans done in cold winter months are more likely to detect brown fat. and it's less common in individuals who are overweight and obese. And so with these 4 covariates, gender, age, outdoor temperature, and BMI, we assembled a propensity score matched cohort where we matched about 5,000 people with brown fat and nearly 10,000 people without brown fat controlled for these variables. And when we linked that to all of the diagnosis codes in these patients' electronic health records, what we saw was quite striking. So I'm showing you here a forest plot with the odds ratios for these different diagnoses. And what you can see at the very top is that there is this really significant reduction in the odds of type 2 diabetes in people with brown fat, even controlled for age, gender, BMI, and outdoor temperature. And we also see significant reductions in the odds of dyslipidemia, coronary artery disease, cerebrovascular disease, congestive heart failure, and hypertension. Now, these are, of course, associations, and they don't necessarily imply causation, but they really provide us with hypotheses to go back and test in our cellular and animal models. The other thing we were able to do is to look at our cohort segregated by BMI. And so we binned people into either normal BMI less than 25, overweight between 25 and 30, and obese BMIs greater than 30. And if you look here for type 2 diabetes, what you can see is that people without brown fat have a steady increase in the prevalence of type 2 diabetes with increasing BMI, and this is very well documented. But what we were quite amazed and pleased to see is that people with brown fat have a significant protection from type 2 diabetes or a reduced incidence at all 3 BMI strata, such that people who are obese and have brown fat have a prevalence of type 2 diabetes similar to those of normal weight who lack brown fat. And we saw a similar pattern for these other comorbid conditions. I don't have time to show you today, but we also looked at laboratory values and things like blood glucose, for example, triglycerides and HDL, all tracked accordingly with brown fat status. And so with these findings, we've moved forward to try and understand what explains these benefits. And I mentioned at the beginning that brown fat is able to convert chemical energy into heat. And so one hypothesis certainly is that brown fat may confer some of its benefits by increased energy expenditure. But many people have been skeptical that this relatively small tissue could really expend enough energy to explain its benefits. And in addition, the benefits I'm showing you are independent of BMI because the study is controlled for body weight. And so we have increasingly considered 2 other hypotheses. The first is, might there be secondary effects on white fat? In other words, might the presence of brown fat promote a more healthy distribution of white fat? And so in a paper that we now have in press, we've shown in this same cohort that when we actually use the CT scans to quantify the amount of subcutaneous and visceral fat, when people are matched for body weight or BMI, the presence of brown fat is associated with less visceral fat and more subcutaneous fat. So it does seem to be that at least one of the ways that brown fat confers its benefits is by promoting a more healthier distribution of fat. And then lastly, perhaps brown fat might convey its benefits via endocrine effects by secreting metabolites and polypeptides that signal to other tissues to regulate whole body metabolism. And so finally, I just want to give you some insights into where we're heading. In looking at our cohort, we were really impressed at the tremendous variation across individuals in brown fat activity. So what I'm showing you here is binned by age, the activity of brown fat measured in subjects in our cohort. And you can see, first of all, that brown fat activity tends to go down with increasing age. But I'm showing you in red all of the individuals with brown fat activity more than 2 standard deviations greater than their age cohort. And you can see a number of individuals who have exceptionally high amounts of brown fat relative to their age matched group. And this at least suggests the possibility that there might be factors like genetics that explain this. And so what we are now doing, really stimulated by the Pathway Award and the support it gave us, is beginning to look at whole exome sequencing data that's available on many of these individuals And then in parallel, we formed collaborations with colleagues who either studied the genetics of thinness or the genetics of obesity to try and identify variants associated with brown fat. And ultimately, as we identify these variants, our hope is that we can study these variants in cellular models and mouse models to identify new genes, pathways, and perhaps drug targets that regulate the function of thermogenic fat. And so in closing, just to summarize, I've shown you today that brown fat allows us to survive key life challenges. And with our modern lifestyle, with abundant energy-dense food, sedentary status, and thermal comfort, that has likely contributed to an overexpansion of white fat and a decrease in brown fat. Brown fat is associated with protection from obesity and cardiometabolic diseases, and a deeper understanding of the biology of this tissue may provide new targets to address obesity and associated diseases like type 2 diabetes. And so just in closing today, I want to thank the people from my group who did this work. This is a picture taken shortly before the pandemic when we had one of our last group outings. And I'm showing you here the way that we interacted for much of the last year. But I also wanted to end on an optimistic note. Now that we're all vaccinated, we recently had our first in-person get-together. And so I just highlight here the names of all of the people involved, our collaborators, our core facilities. And then I really want to again express my gratitude to the Pathway program that made all of this, as well as the subsequent funding opportunities possible. So thank you. And with that, I'm happy to take any questions.
Thank you, Paul. That was really exciting and beautifully presented. Let me ask a quick question first. Do you find the presence of brown fat to be associated with other variables that we commonly associate with obesity, metabolic syndrome, et cetera? And I'm thinking here in particular, is there any association with physical activity or food intake? And the latter is would kind of be logical. If you've got a higher energy expenditure, would you influence food intake? What do you see in human cohorts in that regard?
Right. So unfortunately, we don't have information on either of those things in our cohort. Our cohort was a retrospective cohort, and so there's no data on physical activity or food intake. But we have thought about, with the large number of scans being done, the possibility of initiating a prospective study where it might be possible through wearable devices, for example, to quantify physical activity, and through surveys and other means, perhaps to study food intake. There have been some really provocative links between physical activity and brown fat in mouse models, but I'm not aware of any definitive evidence linking those two in humans.
Would you hazard a guess as in terms of directionality of either variable?
Well, I guess based on the mouse studies, one might predict that physical activity or exercise would result in increased brown fat activity. In terms of food intake, I think that's a little bit more complicated because in rodent models, there's this suggestion that at least early upon exposure to a high calorie diet, there's activation of brown fat through this diet-induced thermogenesis. That's probably part of a protective pathway. But more chronically on a high-fat diet, the activity of brown fat is suppressed. And the fact that we and others see less brown fat prevalence and activity in obese individuals, I think, goes along with that.
Okay, a couple questions here. Marlon Pragnell, great work. Do we know if brown fat levels change in individuals who have undergone bariatric surgery?
Yeah, so there is a very small study from a group in the Netherlands that looked prospectively with PET CT scans in individuals pre and post bariatric surgery, and they did see evidence that brown fat activity increased after bariatric surgery, but it was a very small study, on the order of 10 to 20 individuals.
Okay, and Barbara Kahn, a really interesting work. Can you say more about how the presence of BAT regulates the distribution of white adipose? Is that data only in mice, or is there also BAT regulating WAT distribution in human data?
Yeah, so the data I was referring to in this forthcoming paper are all from humans. And so what we did in our human cohort is we used the CT scans to quantify the amount of subcutaneous and visceral fat. So at this point, we really just have an association, and we're also very intrigued about the mechanism. We would like to think that it's the presence of brown fat that is regulating the distribution of white fat, but it's also possible that the distribution of white fat is actually regulating the prevalence and activity of brown fat. And I think we're really going to have to develop animal models to better assess that.
Okay, and last question from Bob Eckel. Historically, approaches to reduce body weight and related metabolic pathways by increasing energy expenditure have not been successful. Modest effects as well as adverse effects. Assuming that brown fat is increasing energy expenditure, what are your thoughts about this?
Yeah, so I'm very familiar with those studies, I guess, dating back to dinitrophenol and then some other drugs that worked, or drug candidates to increase energy expenditure. My personal belief based on the mouse and human data is that most, or perhaps even all, of the benefits associated with brown fat are not due to energy expenditure, but are actually due to other effects, likely many of them conveyed by metabolites and polypeptides that signal in an autocrine, paracrine, and endocrine fashion. And so that of course remains to be proven. But if that's the case, then I don't think energy expenditure is going to be a key requirement for that.
Outstanding. Thank you for the answers. Very clear. And we'll move then to our last talk of the session. Judith Agudo from the Dana-Farber Cancer Institute in Boston. And she was awarded an Accelerator Award in 2020. And she is representing the critically important area of type 1 diabetes here in our session, harnessing immune privilege mechanisms from stem cells to protect beta cells from immune attack. Judith.
Hi, thank you so much for the invitation. It is really, when you were mentioning that there were more than 700 people that applied and only 34, and out of those 34, I'm one of the ones that get to present. This is really an honor. So thank you for the invitation today. And yes, so my talk is going to be about type 1 diabetes and a little bit of the history that brought me here. And type 1 diabetes, sorry, I guess. So type 1 diabetes, as all you know, is it happens when the immune system by mistake destroys beta cells in the pancreas, which leads to lack of insulin and therefore hyperglycemia. And if we were to cure type 1 diabetes for real, what we would need to do is to replace what is missing. Namely, we need to put back the beta cells that were destroyed. And I have to say that this is actually how I started my, I would say my journey in science, trying to regenerate beta cells. And actually it works. And I mean, I was not the only one. Many labs have done work here. And this is just an example of some of the work I did during my PhD. And this was so exciting to be able to regrow these beta cells, in this case in mouse models. There are other approaches such as transplantation of islets, or many other labs now are also generating beta cells in Petri dish. So it would be as simple as putting back those beta cells that are missing. But this title that it says it can achieve a cure, I would like to change it by could achieve a cure if, which is that very early on during my PhD, I regrew these beta cells, but obviously what happens soon after, I'm sorry, is that these beta cells get attacked by the immune system again. The immune response that killed the beta cells at the beginning comes back and kills the replaced, newly transplanted beta cells. So when this happened, I realized I had to understand the immune system, and I had to find a way to protect these precious beta cells that had been replaced. And with this idea in mind, I left my home country. You can tell by my accent, I'm not American. I came to the US with a Fulbright fellowship, and I went to an immunology lab to learn immunology and to try to find means to protect these beta cells. The first work I did was trying to induce tolerance, but that is easier said than done because the immune system can easily change based on the environment. If there's an infection right now with COVID, if we were to get tolerance in a person and then this person gets COVID, it's likely that that tolerance is going to get lost again. So after this initial work, I realized that for my independent career, if I could, what I would like to do is to find a way to make beta cells invisible to the immune system. So in such a way that the protection is cell autonomous. It doesn't matter what the immune system is doing, they are still protected. And it also has the advantage that you don't have to suppress the immune system at all, because the last thing we want is to have patients with type 1 diabetes being more susceptible to infection or cancer. So with this idea in mind, how one can protect beta cells from immune attack. And this is not a unique idea that I had. Many other labs are trying to achieve this, to engineer either islets or Petri dish generated beta cells so they are invisible to the immune system. And what many of these labs are doing is trying to get ideas from how the immune system works and combine these genes, these knockouts, so many iterations. The problem is there are so many of these iterations that I think it's very hard to get to the right one in a reasonable amount of time. And the very, I think, naive idea I had was, I thought this was an engineering problem. And engineers, sometimes what they do is they copy what they see in nature. So for example, if you want a robot that climbs up a glass, you can try many different calculations and math and engineering things, or you can copy something from nature. For example, I know that there are robots that copy what geckos do, right? They copy the little paws and you just translate that and then it works. So a very naive approach, but this is a little bit what I had in mind. So if we want to protect beta cells from immune attack, what I thought would be the easiest would be to find cells that naturally can escape from the immune system, that evolved to escape from immune attack and copy what they do. And then what we have to do is learn from immune privileged cells. So do they even exist? Do we have cells in our bodies that are immune privileged? And this was already during my PhD that I was reading these reviews about immune privileged cells. And you can find this in many articles and in many reviews that stem cells are immune privileged. So I thought, this is very easy. We just study how stem cells in the body escape from immune attack, and we do the same thing, and we engineer beta cells to do that. But actually, the literature was very, very weak. And if I wanted to understand how stem cells did it, I needed to study them myself. So how do we study immune attack against immune privileged cells, or in this case, in particular, immune privileged stem cells? And the way an immune attack happens, just very briefly for the non-immunologists in the audience, there's this domain immunology that says that all nucleated cells present antigen through MHC class 1. So every cell in our bodies has this machinery that can present self-produced peptides here through MHC class 1, and this is what T cells see. So for example, if this is a beta cell, this could be insulin or any other protein. It's chopped up in little pieces, and this is presented. And then an anti-beta cell T cell, this is what it sees. It binds here and it attacks. So can we study these in cells that are actually resistant to this? And how do we do this? And immunologists, I think I can call myself an immunologist now, the way they study immune responses against any particular cell type is by using what is called a T cell receptor or TCR transgenic mice. In this case, we use T cells for which we know the affinity. We know that the T cell receptor binds X, Y, or Z. So for example, if you wanted to study immune responses against beta cells, you could have a T cell whose TCR binds to insulin presented through MHC class 1, so an insulin-specific T cell. So if I wanted to study immune privileged stem cells, I needed a T cell that recognized stem cells, but that didn't exist, and I needed to make one myself, but how could I do that? There are many different types of stem cells. I wasn't sure which stem cell would be the immune privileged one I wanted to study. And the genes they express are very diverse. So stem cells in the intestine versus the brain versus the muscle, they all express different genes. I couldn't generate a T cell model against every single one of these stem cells. It was not It was simply not feasible. And then is with my mentor back then, Dr. Brian Brown, we had this idea and we realized that actually stem cell biologists, they love GFP, the green fluorescent protein. And there are a lot of mice that express GFP almost anywhere, even non-stem cells that are, right? Like if you just go to Jackson Labs, there are so many mice that express GFP in almost any cell type that you could imagine. So what we thought is, well, if we make a mouse whose T cells recognize GFP, then we can take advantage of this plethora of GFP expressing mice and we can study the immune response specifically against each of these GFP expressing mouse models. Namely, we can study T cell responses against almost virtually any cell type. And then we can find interesting biology. We can find cells that are protected, cells that die, you name it. And this is basically what I did. I got, long story short, I got this mouse whose T cells recognize GFP. And once we had the mouse, this is a mouse that is a just eGFP death-inducing T cell mouse. So we decided to call this mouse the Jedi mouse. Short disclosure, Brian, my mentor, really liked Star Wars. So once we had the mouse, we did the first thing that I did was actually what I knew the best, which was let's try how this works in a response against beta cells. We know beta cells are susceptible to T cell killing. Do these Jedi T cells reproduce this feature? And what we did is we got these mouse insulin promoted GFP mice from Jackson Labs. And when you adoptively transfer these Jedi T cells, what happens is what it has to happen. T cells kill very well beta cells, unfortunately, and these mice develop hyperglycemia. Something that was not so obvious back then, but now is something that we are exploring in my lab, is that thanks to GFP, so first of all, GFP is something that you see without the need of staining. I cannot put into words how many times I had to stain for insulin, right? Like to count beta cell mass during my PhD. Now in my lab, we just take the sections of the pancreas and GFPs there, so we can very easily see beta cells. We can isolate them. And for example, here you can see when these Jedi T cells kill the beta cells in the pancreas, this one guy here is GFP, so it does have the antigen, and this is a Jedi T cell, and it's not killing it. How come is that happening? So now we have the power to isolate this beta cell alive because GFP doesn't need staining, and we can profile and see how that beta cell is doing what, right? Like is surviving in that, I would say, nasty environment. So once we established the model, the question we had was, okay, let's find immune privileged stem cells so we can learn from them. And what I did is I got these mice that Hans Clevers in the Netherlands developed called LGR5 GFP. So these mice express GFP only in stem cells and actually in different types of stem cells, in the intestine, in the ovary, and in the skin. And a very long story, trying to get it very shortly. The first stem cell we studied were the intestinal stem cells. So stem cells that are, the job they play is to replenish the intestinal epithelium. We thought that if anyone had to be immune privileged, the intestinal ones looked very important to us. And actually they are not. So this is how the fax looks like. These are the GFP stem cells. And when you inject T cells, they get killed. So this very big claim that stem cells are immune privileged is not really true. This was very disappointing. What about stem cells in the ovaries, right? Like ovaries are these very special organs, they produce oocytes. So we thought this had to be immune privilege. And this is how they look like by IM, and this is by flow cytometry, the GFP cells. So when you inject Jedi T cells, they don't get killed either. So they were not immune privileged. Okay, so what about stem cells in the skin? This is again how they look like by GFP staining. These are the stem cells in the skin. And here, to our surprise, they did survive, and they actually survived very, 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 very efficiently. They did not get killed. It didn't matter how many T cells we adoptively transferred. These T cells were fully activated. They were completely resistant. So finally, we found what we were looking for. And this was very, very, very exciting. And now these stem cells, I mean, we worked out mechanism, and actually they survive by controlling the antigen presentation machinery in a very special manner. But these stem cells are not only special because they control the antigen presentation machinery, meaning MHC class 1, but these cells were actually described by other labs that they have this unique property of recruiting the regulatory T cells. So if you were to look for Tregs in the skin, they are not random distributed. They are all of them around the stem cells. But it's not known that happened. So I think that we are very interested in my lab is one, how are these stem cells escaping from killing by cytotoxic Jedi T cells, or in general cytotoxic T cells? We know it's through downregulation of the antigen presentation machinery. Two, how do they recruit regulatory T cells? And we want to learn all that. So if we find that one gene that is allowing for this unique property, we can take that and engineer beta cells so they can protect themselves from immune attack. So this is basically the summary. So what the pathway award is allowing us to do right now is to look for stem cells that are immune privileged, understand how they do it. We do have a few candidate genes already, and we use these genes to engineer either primary beta cells or stem cell derived beta cells, or like islets. We engineer them, we put them back in the mice, and we take advantage of our platform in which beta cells express GFP, so we can very easily see them. We can very easily sort them back. We see them alive. And we have a T cell model that recognizes these GFP cells. So we can also induce type 1 diabetes in a very robust manner. And we engineer the beta cells, and we test whether these candidate genes that we have found protect these beta cells from killing. And everything is very preliminary because this started in February 2020, and I don't need to mention what happened in March 2020, but I'm very excited to say that one of the candidates we are testing, it looks very promising, and we see that these beta cells do get protected from T cell attack. And finally, I just have to say thank you to so many people. I have to say thank you first to my PhD advisor, my advisor from my postdoctoral training, Brian Brown, who I already mentioned. Miriam Merat taught me so much immunology. My initial JDRF fellowship to try to find a cure for type 1 diabetes, people in my lab, and then above all, my pathway award mentor, Dr. Andrew Stewart. I have to say that something unique about this award is not only that it funds out of the box science that wouldn't be funded by more conventional sources, it also provides this mentorship that is so valuable. And Andrew has been amazing. He has met with me every time I asked. He actually even assembled a team of people to provide even more feedback to me and to my ideas. I also want to thank Doug Melton, who, thanks to this Pathway Award, my lab has really built up the work in type 1 diabetes farther than I had ever expected, which is very exciting. And Doug saw me presenting, and he liked the ideas I presented, and we also have a collaboration right now. So it's been really, really important for the lab, and so thank you so much.
Thank you so much, Judith, for that lovely presentation. David Harlan asked a question. While great if you can protect stem cell beta cells from immune attack, but what if those stem cell beta cells behave badly? In other words, have a malignant transformation?
Yes, that's always the question. So the approach we are taking, I actually think, I mean, I don't want to sound pretentious, but while other labs are trying to engineer these beta cells, making them knockout for B2M and expressing PD-L1, which is very dangerous because this is actually how cancer cells escape from immune attack, by having PD-L1. The way we are doing it still leaves room for immune detection. So we don't induce PD-L1. We downregulate the antigen presentation machinery, but we don't shut it down completely. And they still respond to some inflammatory signals. So hopefully they would still be able to get detected by the immune system if things go wrong. That being said, with primary beta cells, I don't think you have the problem because it's very rare that a primary beta cell would go into a tumorigenic process. Stem cell derived beta cells, and this is not my expertise at all, they could, but we would then, let's say if with Doug Melton we decide to do something like this, we could always have a system in place to induce a suicide, right? Like a suicide gene in these stem cell derived beta cells.
So in one of your earlier slides, you showed an immune attack on the pancreatic islets and some hardy survivor beta cells, a few hardy survivor beta cells. Is there a path towards understanding how those cells survive, potentially independent of their stem cell lineage, but just understanding how a mature beta cell can be robust in a bad environment?
Yes. So that's actually something I'm tremendously excited about, to now get those beta cells back and understanding how they survive. We see them. Actually, even in patients, you can see them. After many years of type 1 diabetes, you can see patients with beta cells surviving there. We do see that, for example, MHC class 1 is very low in these cells. We don't know how they are able to keep it so low in this inflammatory environment. This could be one way they are escaping. But yeah, we are just starting that and we are very excited.
Great. And David Harlan, again, if you transplant the skin EGFP stem cells, do they also in that setting resist Jedi killing?
Oh, that's a very good question. So I did not do the transplantation. I did an in vitro experiment, and in vitro they still survive. But so we do think, so we think it's a cell autonomous process and it's not controlled by the environment because in vitro is retained. But yes, I did not put those stem cells in another place. They are very tricky. And again, I'm an immunologist. So every time I worked with a stem cell, I was not an expert in that cell type. So transplanting them was not the easiest thing for me to do.
All right, I'm not seeing any more questions in the Q&A. And we're coming to the top of the hour, the end of our program. So let me just take a moment to thank the 4 speakers today for representing the program so well. Really want to have a shout out to everybody who's been an awardee. We've been doing a monthly research update, and to my view, the quality of all the presentations has been outstanding. So, I mean, I'm not kidding when I said there could be anybody of the 34 awardees could have done a great job representing today, but that shouldn't detract from 4 wonderful talks that we heard. For those of you who are not members of the Mentor Advisory Group or awardees that have attended today, I'll just conclude again by saying that in my 30 years of association with the ADA, I have found the Pathways program to be really one of the most robust and impactful programs that I've ever seen in the ADA repertoire. And if so moved, the contributions to this program would be deeply appreciated and will manifest in the kind of creative, out of the box kind of science that you heard today. And that's our hope and that's our wish. And to all of you, have a great rest of the ADA meeting. And again, thanks so much to our presenters today. Thank you all very much.